Okay, so I've been quite interested, as you just heard, in cortical basal ganglia connectivity. And more recently, we've branched beyond just the cortical basal ganglia connectivity, but to also cortical connectivity. And one of our interests, the driving interests, have always been to understand better the circuits that underline motivation and cognitive behaviors. And so even though I worked in Parkinson's disease, it always was with an idea of the non-motor aspects of Parkinson's disease, because as you probably know, Parkinson's is not only a motor disease, but also affects mental health in many different ways. And so the idea, though, that has driven a lot of my work is trying to think about the brain across different kinds of networks. We have a sense or a tendency to clump lots of different functions together, and I do too. But I've been quite interested for quite some time in understanding, in particular, how the reward and motivation systems interact with the cognitive systems. And this started in the basal ganglia for me because, of course, the basal ganglia has always been divided into these three areas: cognitive, limbic, and motor. And yet, when we behave in the world, we don't just tap into one and not the other. So our interest has very much been driven by this idea of where do the connections across these circuits go. And so just to start, this is just a snapshot of various parts of cortex and striatum and basal ganglia, and the functions that we pin on those based on a collection of lots of different studies. So weight behaving primate physiology studies, human imaging studies, and so on. And of course, each area is its own area of research. And I'm just giving you a soundbite of some of the things that people link to these. So the VMPFC is important in chosen value. The orbital prefrontal cortex links stimuli to their values. Goal-directed behaviors is the focus of the research in that region. The dorsal anterior cingulate just does just about everything. So, but I think guiding action switching is one of the main components that people think about. The dorsolateral and the ventral lateral prefrontal cortex are involved in cognitive control and attentional elements. And we know we always link the ventral striatum with reward and motivation, and the dorsal rostral striatum with cognitive control. So these are kind of the areas that we think about in terms of what functions they have. And of course, all of these areas are linked to abnormalities in many different psychiatric diseases, in particular OCD, depression, schizophrenia, addiction, PTSD, et cetera. And in fact, if you drill down to imaging studies and white matter studies of these diseases, the imaging studies will always give you a collection of these areas that are abnormal or changes. And as I said, I think that this is just an example of how we actually think about the brain. These are resting state networks that have been described, the well-known default network, and parcellating out the cortical networks into these large areas, which are sort of data-driven networks, which are then lumped together in particular functions. But they're large regions. And then likewise, the cortical basal ganglia system has always also been thought about as being separate and segregated, with the limbic and the cognitive areas linked to specific cortical regions, and not on here the motor areas, which of course have their own set of connections. So we've been interested, as I mentioned, is, how do you really go from being hungry to wanting a pizza to thinking about where you're going to get the pizza to actually the action of getting the pizza, all of which is going on in your head at kind of the same time. And some time ago, we published a paper that, and I'm not going to go into this now, but basically it was demonstrated that one way in which the limbic components of the basal ganglia can influence the more dorsal cognitive and motor components of the basal ganglia is through these connections through the substantia nigra and the dopamine neurons, where the limbic areas project to a region that projects back up to a more dorsal area of the striatum. And through this series of loops or spirals, the more limbic regions or the motivational regions can influence the more cognitive and motor regions. And that subsequently has been demonstrated in a number of different behavioral studies to exist by other labs. But we've now been focused on two things. One is, this is assuming that the cortical striatal connection is fairly straightforward and topographic. But some of our more recent work has demonstrated, well, maybe that's not entirely the case. Maybe there's actually integration going across these functional regions within the striatum. And in addition, we've also been interested in regions of the cortex that crosstalk between these areas. And you probably are aware that there's a growing interest now in how those broad cortical networks that I just showed you before are actually connected with each other. So we have these broad distributed networks that have some kind of function, but how do they actually talk to each other? And one idea is that there exists hubs, which is kind of taken from the graph theory ideas, that really integrate and distribute the information across these networks. So what we were interested in is how do we actually demonstrate some of these things from an anatomical perspective. The other area I want to talk about is the white matter. The white matter, which matters, and we've known about connections between the white matter, for hundreds of years, right? Not hundreds, but, quite a while. But the information between white matter, or the interest in white matter, really got dropped for decades until diffusion imaging came along, which was actually really beautifully demonstrates so many pathways. And just to give you an idea of this is just a graph over the last 5 years of white matter papers that have come out. I mean, there's just this huge, and that's not surprising to you, I'm sure. And so this is just showing you very briefly the three pathways, which I'm going to talk a little bit about: the corpus callosum, the cingulum bundle, and the internal capsule, are areas that have really captured many people's attention, especially because of the FA changes in a number of diseases. And over here, I just have a series of mental health diseases that have focused on the white matter, and part of this literature that has extended so much. So what I'd like to do then today is it's divided into two parts. One is the connections between regions, gray matter and regions, and white matter. And so just sort of briefly, we work in the non-human primate, and we use really standard, very old, traditional techniques. There's nothing whiz-wang about what we do. They're just tracer injections that we've been doing for, again, decades. I hate to say that, but it's true. And so here you have a monkey and a brain, and here you can see the fibers. You can just visualize those fibers coming up from an injection site. And the goal here is, how do we go from the precision of the anatomy that we can see to the indirect methods that we have of looking at connectivity, both using diffusion imaging, but also functional MRI. How do we translate from this to this? And then how do we actually back translate to a rodent? And because I know there's a great interest here in the marmoset, also obviously an outstanding model. And so how do we take what we know here and come in both directions? And the non-human old world primate is a really good translational piece to go in both directions. And then taking what we know from these areas, of course, drilling down to understand disease. Okay, so just a couple slides on methods. So here's an injection site. Here's a coronal section of a non-human primate brain. You can kind of see the striatum here. It's not counterstained. You just see the injection. You can see the beautiful pathway that's coming out, the fibers that are coming out from that injection site, which is blown up here. In addition, this particular tracer type gives you both anterior and retrograde. So here you can see the nice labeled patches in the striatum, and also the labeled cells in various parts of cortex. So some of the tracers are unique to be anterograde, some are unique to be retrograde, and some are both. And this is just to give you an idea of our ejection sites in cortex, in the prefrontal cortex. And you can see we've pretty much covered most of the orbital and most of the medial, the ventral medial, the anterior cingulate and the dorsal medial, and much of the lateral cortex. So we really have injection sites throughout there, so we can really understand how this system, how the organization works. And then what we do is with each one of these injection sites, we can outline the pathways. And here's an injection site with the pathways. You'll see a little bit more of this later. So these are the white matter pathways and the terminal fields. So for each one of these injection sites, we can map out the complete projection and their white matter pathways and how they get to where they're going. And then we can combine them again in a 3D model. And so this is it would be too complicated to do more, but this is just showing you 3 different injection sites in 3 different colors. And so that we can, I don't know, I guess the light is shining on this, so you can't see it that well. But what you can then do is take all of these injection sites and compare the white matter pathways with each other and understand their organization. And you can understand the organization of the terminal fields with each other. So this is just outlining what we're going to be doing. So we take the anatomy studies and we determine the location of the hubs and the distribution of those and the pathways, the white matter pathways in the monkey. And then we can see the analogous region in the monkey diffusion MRI. So along with our collaborators at the Martino Center, we have imaged many of these monkeys. So there are about 15 of those that have a high resolution diffusion MRI, so that we can actually compare that to our anatomical pathways to the diffusion. So you can really see which, what is actually a technical issue and what is a real pathway in the diffusion. And then we can translate that to the human, both diffusion MRI, and then we're also working with colleagues looking at the functional MRI to understand those connectivity patterns that we see in humans. We're also using tracers, and I'll show you a little bit about that, to understand the rodent brain, to, again, identify the homologous regions that might be hubs and their pathways to, again, probe that system in more detail. And then finally, we can compare disease with healthy control subjects to think about where those disease connections are abnormal and how we might probe them for, use them for targeting. So I'm going to start first with the cortical basal ganglia system. And again, this is just what you see up on the top are examples of one injection site in the VMPFC, OFC, and the dorsal anterior cingulate. And what you can see is their projection from each of these, and I picked them specifically to show that they're somewhat separate from each other. However, you saw how many injections we have in each of those areas. So when you combine those areas without overlapping another area, right? So all of them have to be within, say, the VMPFC and not the OFC, and within the OFC, and et cetera, what you end up is with obviously a much larger and broader projection region. So this is VMPFC. This is VMPFC plus OFC, and just the dorsal anterior cingulate. And the reason this is separate is that one of our findings, interestingly, is that all cortical areas are not created equal in terms of how they project to the striatum, and that even though there's a topography to this, which you can see here, the dorsal anterior cingulate is a kind of special region because even with the same relative size of the injection site, it seems to impact on a larger area of the striatum than most other cortical regions, and you can see that here. If you put them all together, you get a picture that looks something like this within the striatum. Now, if you think about what people think about as the ventral striatum, that's the area that's associated with reward and motivation. And usually what you'll do is you'll divide it right here, something like this. This is usually your typical ventral striatum. However, there's not one of these projections that's limited to that area. So right away, you can see the schematics that we have come to use all the time may not be as precise as we would like, especially if you're looking at imaging studies. So if you find an area that is responding to some kind of reward, and you find that area up here, well, you could probably still say it's in the ventral striatum, even though it's not ventral. But if you define the ventral striatum by virtue of its inputs from some of these regions that are associated with emotion, then it is well within that. And so you might start thinking about how you put your seeds into an area to probe it, and also how you analyze those. So again, this is putting them together. And what the other point that's important here is that based on this, you can see that the striatum, and we've now validated that with some collaborators, that it's very similar in the human, that 20 to 25% of the striatum, the whole striatum, is occupied by this sort of limbic or emotion reward area. So that's a very large area. And also you can see this is rendered in 3D, and you can see that each area, although topographically organized, there are lots of little spots in there in which there's a convergence of various kinds of inputs. So what does that convergence look like? If you add the dorsolateral prefrontal cortex, you have a similar kind. I added it in blue just so that you could see the differences between there. But you see that things are very, very mixed. And then we went back, translated again into what does this look like in the rodent? And we found that, in fact, the rodent has a very, very similar kind of organization. That is, there is a topography, but from IL, OFC, PL, and anterior cingulate, you still see this kind of a mixture. So there's topography, but within that topography, there are clearly areas in which regions converge. And it's those areas that may be interesting to probe for specific kinds of behaviors, rather than just simply say we're now in the limbic versus cognitive systems. With Bruno Everback at NIH, we then looked at a probabilistic map, created a probabilistic map of all of our injections. Actually, at the time, there were relatively much fewer than we have now, so we're going to redo this map. But basically, what we found was that this region right about here is an area that gets input from all 5 of those areas that I was just talking about: VMPFC, OFC, dorsal lateral, ventral lateral, anterior cingulate. And so we think that this could be, right, a kind of a hub, because these are areas that are coming together that are not normally part of the same network. We also had a retrograde tracer. It just so happened in this region up here, and we had 4 out of the 5, and the 5th one that was not included was the VMPFC. Here are the cell counts that we stereologically counted those cells. You can see that VMPFC is pretty much left out, but orbital, ventral lateral, dorsolateral, anterior cingulate are all there. But in addition, we had caudal cingulate cortex and a part of parietal cortex. And this is an area in parietal cortex that is a perceptual area, and an area that is actually activated when an addicted individual is in a scanner and shown drug cues and start to crave. So this is a perceptual area that, is very, seems to be linked to many things, of course, but addiction is one of them. And so Eunyoung Cho in the lab then did an analysis of the inputs in the striatum, and this is all based on the striatal anatomy, where she saw that here in this region, as we might have predicted using anterior grade projections to the striatum, a hot spot in that area. And then she probed that area using resting state MRI in the human here, and then control just lateral to and a little bit ventral to that seed, and found that this seed very much was very similar to the inputs she got from here, which included the area of parietal, dorsal lateral prefrontal, ventral lateral prefrontal cortex, and she also got some orbital in this, and the anterior cingulate, whereas the seed just placed a little bit lateral and a little ventral got a much more limited input. So these are ways in which we're thinking that we can use these kinds of maps to go back into the imaging world and probe those networks more specifically. Again, back translating it, going back to the rodent, we want to ask the question, should we start thinking about reparcellating out the rodent striatum? And so Sarah Hollenbrenner in our lab then came up with the very clever idea of starting with the homologous regions that we know are homologous across species. So we know that the amygdala is the amygdala in the human, monkey, and the rodent. We know that the shell of the nucleus accumbens, we can identify it over all three species, and we know that we all have a hippocampus. So she asked the question, let's outline those areas in the striatum. So this is the shell, this is the projection region from the hippocampus, and this is the projection region from the amygdala in the rodent and in the monkey, okay? And right away you can see that there is a really nice homology. They're kind of positioned in very, very similar places. What does that mean? What that means is that we can now ask what projects to those regions. What projects specifically to those regions, what projects partly to them and partly outside of those regions, and what is the interaction between areas that project to these regions and outside that area. So starting with IL in area 25, she found that they mainly occupy this whole region, which is pretty much outlined in the amygdala projection area. And then she found that PL in area 32 partly project there, but also partly outside of it. And area 24 and anterior cingulate in the rodent is a problem. So that's something that we are starting to think about, how we divide the rodent and the human cingulate in more detail. And the same thing was true of orbital cortex. Nobody really had a good idea of the homologies between the two. And using this method, you can very nicely see that the areas that are occupied by MO and medial orbital and what's called VLO in the rat and the central and lateral orbital in the monkey have very similar positions. So again, using this kind of method, we can start thinking about how we might divide up the rodent. And then based on that, there are two take-home messages here in the slide. The first is that you can see that the color coding here, has some real similarities, which is really interesting. And you can see that I showed you that the ventral part, the medial, the sort of limbic reward motivation region crawls up the medial part of the caudate, and you can see it does the same thing in the rodent when we compare them in this way. The orbital areas occupy a similar place, and we have some of the anterior cingulate also in orbital in the same areas more laterally. The other take-home message is that each of these areas don't get input from one region. Again, similar to what we saw in the monkeys. So what's in large letters are the main inputs, and then the smaller letters are also inputs, but are not the big dense inputs. So again, I think coming back to this, where there's a lot of work we're still doing on this. One thing that we've realized is the PL in the rodent. Our new data, as well as the data of Greg Quirk, who is a collaborator of ours in our Conti grant, are starting to think about the PL as a very complicated area and probably should be divided into 4 regions, certainly a rostral and a caudal region. And if we do that, my guess is, and we're thinking that the PL part, the rostral PL, will be more similar to area 32, and that the caudal might have elements of anterior cingulate in the monkey. So let me switch now to the cortex, and so again, if we think about regions of cortex, there's this reward motivation region that most people will agree VMPFC and OFC are part of. There's a cognitive area with dorsolateral prefrontal cortex and ventrolateral prefrontal being part of, and there's a motor area. And yet there are regions like the dorsal anterior cingulate and VL and dorsolateral prefrontal cortex and probably pre-SMA that really have a foot in more than one of these networks. And so what I'm going to do now is just focus on the dorsal anterior cingulate cortex. I'm going to play this very brief movie. And what this is going to show you is, so remember I mentioned that we can look at the projections from these different areas of cortex to the cortex. So these are some of the injection sites. Again, there are only a few. We have just started this project. We have many more to go through. But these are the injection sites in VMPFC, orbital, dorsolateral, prefrontal cortex, ventrolateral, prefrontal cortex, et cetera, anterior cingulate. And what you're going to do, and also the amygdala. And so what you're going to do now is see the projections, which are indicated by little stippling from each of these injection sites. And what you first notice is they pretty much stay in their own area, but many of them spread. There's the anterior cingulate. You can see how spread out that is. From this one VLPFC, you can see it also spreads. Dorsolateral prefrontal cortex. And then the amygdala. And so taken from this, we started to look at these maps and ask questions, where do a lot of these projections come together? And the one that we started with is an area in the anterior cingulate, in this region of the anterior cingulate. And so if you look at the literature of the many anatomists, outstanding anatomists that have identified the inputs to the anterior cingulate. Here's a little schematic of the anterior cingulate, which this is kind of off. It should be over here, but oh, it's also weird, but okay. These are all the areas based on the anatomical maps that have already been done that project to the anterior cingulate. So there's really quite a collection of regions that have been shown to project there. But they can project in two ways. And we know from the literature, actually, there's a kind of a gradient from sort of more limbic reward regions through associative through motor regions that go in from the ventral caudal, moving in this direction, back dorsal and caudal, so ventral moving in that direction. So the other possibility, which is what we wanted to ask, is whether there are areas within that gradient that stand out, that are anomalies from that gradient. And Wei Tang in the lab has been exploring that. Here are injection sites that we have in the anterior cingulate. This is white matter, so the cortex would be surrounding this, just so you can see the injection site. And she's been exploring that. And remember, that's the map we've seen from all the inputs from the literature. And here are injection sites. We've numbered them 1, 2, 3, 4, 5, 6 to go around in this manner. And ask the question whether or not any of these injection sites stand out as outside this gradient. So we have a lot of work to do on this, but basically the three on the bottom, this is the pattern of projections laid on to all the projections that have been shown in the literature, again, which we can validate that we see all those projections. And in red for each of these indicates 75% of the projections come from these regions to that injection. So here's injection 1, 75% of that comes from these areas in red. And you can see there's a big similarity between these. They're in this more rostral and dorsal one, you start to pull in a few other regions, but it's pretty much reward motivation kind of related. And then if you look at the 4, 5, 6, you see that you're starting to pull in more. And this one is much more cognitive and motor related, premotor related. This is what we have identified right now. You can see that many more branches are labeled in this than in the others, and picking up areas that are not necessarily represented in the others. And this is the region that we now believe may serve as a hub. And Wei is now drilling down even further. We have a few injection sites flanking this to actually make sure that this is true. But it is an area that we find in terms of functional imaging that seems to be very commonly seen in a number of diseases if you look at the homologies across species. She's started to look using monkey diffusion tractography to ask the question, where do the streamlines most converge? And again, she's getting a pattern very similar to what we see in the anatomy in terms of the strength of the inputs to that area. And then finally, looking using the connectome data, we find an area very similar to what we see in the monkey in this region of the anterior cingulate. And unlike the monkey, we see also an area more caudally. But we find a region in this rostral part, again, an area that's been demonstrated to be activated in a number of diseases. With Haixing Liu at the Martinez, he's also now started to probe this using resting state, again, by very precisely seeding the regions that should project there, and again, having a very nice correspondence with this region in the dorsal anterior cingulate. So just as a summary of this part of the talk, I just wanted to summarize here by saying there's crosstalk between reward and cognitive control networks that we see in both the striatal and cortical regions. In particular, I showed you the dorsal anterior cingulate. And we think that these areas might be specifically important for changing or adapting behaviors appropriately. The hope is that in a rodent model or perhaps in the marmoset, one could then identify some of these areas and probe more deeply as to what kind of the microcircuitry that underlies these complex inputs in these regions and how they may behave, may be manipulated in behaviors. And again, thinking about them as possible therapeutic targets, and I already mentioned that point. So I want to switch now for the last 20 minutes or so to the white matter pathways. And the three I'm going to kind of touch on, I'll speak most about the anterior limb of the internal capsule, but also work we've done on the cingulum bundle and the corpus callosum. And so these areas, as I mentioned, are the ones that are most prominently that people find abnormalities in many of the psychiatric diseases. So what we had done, Julia Lehman in the lab, we first actually started this by using injection sites and just probing the ventral surface of the brain, the ventral medial prefrontal cortex and the orbital cortex, and just ask the question, how do fibers get to where they're going? And is there an organization to this? And so what we found was, in fact, there are a number of points that we found very consistent across the white matter pathways. And you'll see this again, but the white matter comes out of a cortical region. They divide into very specific bundles that carry specific information and then travel to their targets. And I don't want to go through all of this. It's very confusing, but I do have a little movie to show you, to give you some perspective that these areas, if you do these injections across the ventral surface, they have a particular projection organization. So this is the uns and fasciculus. They come over and cross the corpus callosum. You can see they're ordered. They go through the cingulum bundle. And so you can see the corpus callosum has a specific organization to it. The internal capsule, again, you can see a specific organization, a ventral to dorsal organization. And then this is going to just show you the, for perspectives, that's the anterior anterior commissure. So in these fibers are also descending into the brainstem. But what the take-home message here is that these three different colors are representing three different pathways that are not that, injection sites that are not that far apart, and yet they have a very specific organization. And so to be clear about that, here are the injection sites. There were 4 of them, 4 examples of them. And what you can see is in the internal capsule, we can tell how they enter the internal capsule. So if you're in this very medial part, you enter from the ventral surface. If you're more lateral, you actually enter from the dorsal surface. So you're entering the capsule in a different way. And then you live in the capsule in a different way. So if you are more medial, you live ventral. If you're more lateral, you live more dorsal. And all that is telling us is that there are rules in which fibers get to where they're going and how they are positioned within a pathway. And so why is that interesting? Because when you look at FA values, for example, in disease, changes in that, they're never throughout a whole pathway, but they're always spotty. So the question is, what do those spots represent? And what we're hoping for is to be able to be more precise about abnormalities, white matter abnormalities, in terms of what kinds of connections we're talking about. And so these are photomicrographs showing you the medial orbital fibers embedded in the internal anterior commissure and beneath it, and the more dorsal ones, and from the orbital, and then the more lateral ones. And so then I had made a trip to Oxford, and I gave a talk, and I was really excited to meet Tim Behrens and the Oxford group, and from that came a great collaboration in which we started to ask the question, well, can we see these things using diffusion imaging? And in fact, we could see many of these things using diffusion imaging. So this is the monkey histology, the monkey model that I just showed you based on the histology, monkey diffusion in a different monkey, not the same, and human. And we saw many of the same features, but some were not able to see. So, so I came back and we continued that collaboration, but then I had the fortune to also be a visitor at the Martinos and met Anastasia I never can say your last name right, but Yandeki, I can say it when I'm not in front of a people, but that's true. And so she and I have now been also exploring this in detail. And so what we've done is we started to image some of our animals. So this is where we now have imaging in the same animal as we have the injection. But what I want to talk about first, before I get to that, because this is really key, is that what you know from the pathways, here's this beautiful pathway I showed you before, it's coming from here. This is the injection that's coming from the ventral part, this injection is coming from the lateral, and this is coming from the dorsal. And these are the streamlines, I'm sorry, not streamlines, these are actually the axons. So these axons are here, and these axons are coming in here, and these axons are coming up. And so what you can do is you can very precisely see them, and you can very precisely draw them, and then you can put them in a 3D model. And how they get to where they're going really depends, of course, on how they leave the injection site. So what we found was that when they come down, we're calling it it's a stalk that comes down. It divides into 3 things. This stalk does not really contain a lot of cortical fibers. They're kind of peeling off. What the stalk contains is, and when they divide, is the corpus callosum, striatal fibers, and capsule fibers, so internal, external, extreme capsules. And all of these will show that division. And the reason I'm paying a little bit of attention to this is that this is one place that diffusion can go wrong. So I'm going to first show you the internal capsule, and to show you all of our injections. Again, what you can see is this is the one you saw before, but now there are many other regions. There's the orbital, the anterior cingulate, the ventral lateral prefrontal cortex, and the dorsal lateral prefrontal cortex. And what you can see is that, again, as I mentioned for the pathways I showed you before, they're very well organized. You see a nice topography. So we wanted to outline that topography in more detail. But first we had to make sure that we could take our diffusion, our anatomy into the diffusion. So here's an injection site. Here's an animal that had, this is the same animal, so it's diffusion in the same animal. You see the injection site, beautiful injection site. You see the streamlines shooting out, the axons shooting out, the streamlines shooting out, corpus callosum, really, really nice. However, what you can see down here is where diffusion is not replicating correctly, and you have false positives. So you see the false positive here in the fornix, and there are a number of other areas that have false positive. So we asked first, can diffusion divide into these three areas the way we see it in the actual tissue? And so what we found, and this is this work has also been helped by Georgia, a graduate student of Anastasia's and mine. And so what we see is that you can identify the correct pathway and you can identify an incorrect streamline. Yellow is correct and the blue is an incorrect streamline. And once you can do that and you understand where they go off, you can start to do that in the human. And the reason it's going off here is what happens is, for some reason, we find that the streamline also goes down, sends a false positive, and I don't want to go into the details, into the striatum. And so here you can see in a human, once you know that, you know the one to follow. So in this case, you can see that this is a streamline that's correct. It's coming down. This is going to divide into the internal capsule and into the extreme capsules, but this one is incorrect, this blue one. And so using this kind, and it is in 3D, here's the correct one, and here's an incorrect one. So using this kind of information, we can now ask the question, how is the internal capsule organized? How is it organized? Of course, we first identified it in the anatomy in the human, in the monkey, in the diffusion in the monkey, and then in the human, by eliminating the false positives. And so this is just illustrating how well we can replicate it. This is, again, based on the anatomy, the nice topography. It's a dorsal ventral rule. I won't go through all the rules, but if you're dorsal, you'll be dorsal in the internal capsule, middle, and ventral, and you'll have that same organization within the internal capsule. You see the diffusion gives us the same pattern in the monkey, and the same pattern in the human. You do have the false positive, but we know this one's probably the correct one. So again, we can replicate that in the human, understanding that, and then we can go back and actually look at individual cases using the connectome, and you can see that in individual cases, they're located in a little bit different places, but you can see the pattern is similar throughout the human. So what does that mean? So this is how the anterior limb of the internal capsule is then organized. You have a dorsal ventral, a medial lateral rule. There's some overlap given the compression of the capsule. But by doing this, we have some good idea here now of which fibers are actually passing through these regions. We can make an FA map, which we did. This is a heat map and run statistics on the different components and see that each of these areas, this is a normal brain, actually are statistically significantly different, that the FA values to these areas that carry these fibers are different. And then with Mary Phillips, also part of our Conte, she then probed this using our map and asked the question, are the abnormalities that we see in the CHICs in bipolar disorder patients in the anterior limb of the internal capsule, can they be associated with a specific set of pathways? And indeed, she did find there was a significant difference between the FA values specifically in regions that carry the VLPFC fibers. And that was very exciting for her because she had already shown in a number of other situations, a number of other papers, the activity in the VLPFC was abnormal in the VLPFC. And this is a map that was generated by another collaborator, Rita Goldstein, using cocaine addicted individuals and looking at their FA map. And again, we haven't really analyzed this, so this is very, very preliminary. But the idea, again, is this is a typical map you might see of FA changes in disease, whereas I mentioned, you have a very spotty organization. And looking at this, one might say, and again, we haven't really explored this, so this is something that's just super preliminary, but if you look at this, you might say, well, just maybe the VMPFC fibers, there may not be any abnormalities there, but in their connections, but we could start to look closer at the VLPFC, the DFC, dorsal anterior cingulate, and the OFC fibers. So these are just ways in which we can use these maps. So just an example to now look at diseases. The other use of this, of course, is looking at deep brain stimulation. And so we've been also working with a number of groups looking at deep brain stimulation. And here are the electrodes for the deep brain stimulation of the internal capsule. So here are the 4 electrodes. This is for the anterior electrodes for the ventral striatum, again, for contact points. And the Helen Mayberg electrode sites for the depression and for the deep brain stimulation for Parkinson's disease and for OCD in the STN. So I want to just kind of mention a few of these ways of looking at these as well. So for the deep brain stimulation in the internal capsule, you can see in our model, we can put the electrode in a model of these fiber pathways that I just showed you and start to ask questions about when you stimulate each of these contacts, which fibers, which connections are you likely to be activating. And then what we've been doing is taking patients that have the electrodes implanted and merging a CT scan that identifies the contacts with their MRI scan, a high-resolution structural MRI and a diffusion MRI, and then place seeds in the diffusion. We're merging those images, place seeds in the internal capsule at the contact sites and ask questions about, given the parameters that they're being stimulated and the particular contacts they are using in that individual, can we identify the likely pathways that are going to be activated? And what we found is then they're just beginning to break the blind of this particular study, so I don't really have the results. But what we do know, and this makes complete sense in terms of the anatomy, which was that the lower the contact was and the more caudal it was, the more anxiety was produced in the patient. And so they didn't use that contact, but stimulated a more dorsal one. And when we looked at the patient's outcomes in terms of their imaging, we found that indeed the lower, more caudal contacts were ones that were projecting both to partly through the hypothalamus and to the amygdala. So when we moved those electrodes, the contacts stimulation more dorsally, and then in subsequent patients a little bit more rostrally, we were able to get a better outcome. So this is how the anatomy can help inform these kinds of therapeutic targets and actually change them. So again, just not to focus only on the human and the monkey, we've also started to probe what does the internal capsule look like in the rodent. Again, because the internal capsule had not been described very well in the rodent. In fact, the internal capsule in the rodent is usually considered after the anterior commissure, and the fascicles that run through the striatum were not really identified in an organized way. And without going through a lot of the detail, what we found was that these fascicles within the striatum of the rodent are in part the internal capsule, the anterior limb of the internal capsule. So these are the fibers that are going from the areas of cortex to the thalamus. And what we found also is that they're organized in a very similar manner as we see in the in the monkey. And so what that allows you to do, so there's a nice topography, both in the rostral areas where there isn't a clear internal capsule, as well as in the more caudal areas where there are. And what that allows us to do then in the rodent is be more precise if somebody is wants to do a stimulation experiment. Now we have an idea about what fibers they're going to be stimulating. If you're doing a lesion experiment, you want to be careful because you're not only lesioning, it depends on the kind of lesion, but you might want to be careful because there may be fibers that are actually not part of the striatum that you're affecting. And it also allows us to, for those that are doing cell slice preparation and they want to capture the cortical thalamic projections, they now can, we now can, in our 3D model, provide the angle that is best suited for developing that. So there are a lot of ways that we can use now the rodent model to explore things that we weren't able to before, just knowing where those fibers are going. The other two pathways that I'm going to briefly talk about is the cingulum bundle and the corpus callosum. So the cingulum bundle is an interesting bundle, and we think about the cingulum bundle as just connecting the cingulate cortex. And we know that fibers run in the cingulum bundle that connect to the cingulate cortex. We don't usually think of the cingulum bundle as a highway that fibers might use that have nothing to do with the cingulate cortex. But in fact, that's what we found. We found that fibers from the ventral, for example, from ventral medial prefrontal cortex might hop on the cingulum bundle and then just hop off at the dorsal medial prefrontal cortex or the dorsal lateral prefrontal cortex and just use it as a pathway. And so that's quite interesting, again, in terms of thinking about when we look at abnormalities in the cingulum bundle. It's one of the most prominent fiber bundles that we see FA changes in. Again, where they are within the bundle is important in identifying what connectivity profiles you're looking at in terms of where changes might be made. And so this is a paper by Sarah again, who identified the areas of the cingulum bundle and what goes through them. And so they're color-coded here in green, but the main thing that I want to point out is that this section, this little pink section here, has a number of fibers that are running through it that don't run through it here or here. And in particular, this region carries VLPFC fibers and lateral orbital fibers, which don't run through any of the other areas. And so we can make maps, again, of what goes where. And the important part of this is, again, looking at therapeutic targets over on these two images. Cingulotomies, as you probably know, is still a procedure that is often used for treatment-resistant depression and treatment-resistant OCD. And actually, they're carried out here at Mass General primarily. Ahmad Eskandar is one of the main surgeons doing them. And here you have an idea about where the lesion is. Here's an older lesion. They've changed them a little bit, but you can see that their cingulotomies are going to include not only the cingulate cortex, but of course the cingulum bundle. And what we know is that this area is sort of in between these two. And the interesting thing is that again, we're starting to probe this because now we are getting the patient's images from individuals that have had cingulotomy. And so we want to probe and ask the question whether or not it's better to interrupt fibers in this region that have the VLPFC and orbital fibers, or if you want to avoid that region, right? So these are the kinds of things we can now ask now that we understand how these fibers come through this area. And again, this is just some preliminary work that we've started to look at one patient that does have a cingulotomy lesion, and you can see it right here. It's interrupting these fibers, and you can see it here. It's interrupting fibers from the caudal part. The last area I want to talk about is the corpus callosum. Again, a fiber pathway we all know very well. We all know that it connects the two hemispheres. And in fact, for a long time, we've known actually the parts of it. And we've also known for a long time its functional organization, really, because it's just connecting the two sides of the hemisphere right at that point. So if you're in motor cortex, you know, it's going to be carrying motor cortical fibers to the other side. It's fairly straightforward. But this part carries the prefrontal cortex, and it's very large because the genu, you know, comes down. It's a very, very large region. And so we asked the question, how is this area organized? And you can see that we found, as I mentioned, as you saw in the little movie, it's organized really, really very well. And so we've been now looking at many of our injections. This is just the kind of pretty picture of our injection sites, all the fibers funneling through to the corpus callosum and to this part, because this is the area, because when you think about it, all of the areas before the corpus callosum have to travel back, cross, and go forward again. So in this region, you have all of the prefrontal fibers all kind of smashed together, but they're in a very thick dorsal ventral organization. And so you can see there is an organization to this. I don't know if you can see this very well, but again, according to color-coded, according to cortical region, again, another way to think about abnormalities in disease. So in summary of this part, I just wanted to say that we have similar organization through the anterior limb of the internal capsule and the corpus callosum. And the rodent, we can identify an anterior limb in the rodent. It has a similar organization. And we can segment these big bundles according to specific connections, and we can use those to link with disease, which allows us to have a better idea of how we target those diseases. But I do want to say that one of the things that's driven this so far really is the ability to use the anatomy of the monkey to understand what we're seeing in diffusion and what we're seeing in functional MRI. And so again, just to end with the slide I started with, so we're using this very precise ability to identify pathways and connections to understand the human brain, to back translate to the rodent and to the, hopefully, marmoset, and to understand human disease and targets. And with that, I'd like to thank the people that have really helped in all of this. These are some of the people in the lab that I've identified with their work. In yellow are the members of my Conti organization. I want to highlight Anastasia, who's really been so important in helping us think about the diffusion, and Tim Behrens and his group as well, and Haixing Liu, who's really been so helpful in thinking about functional connectivity profiles. So thank you very much. Grab some water.
You'll be happy to answer any questions. Do you know her? Behavior.
Well, I think that understanding how connectivity profiles interact does help us understand what parts. I mean, it's a two-way street, right? So we all get information from behavioral studies and how emotions are derived, and then we think about areas that might be connected to develop those kinds of behaviors. So it certainly drives my work, and my guess is that people who study those areas also think about the structures that we find are connected.
Are
you actually are they actually trying where is she stimulating?
Right, so when deep brain stimulation started, it was assumed that you were stimulating the, oh, that's my timer. It was assumed that you were stimulating the gray matter, and you're stimulating the structure that you thought was involved in whatever it was. But we now know what the stimulator parameters people use, it's mainly the white matter. It's mainly the myelinated fibers that you're stimulating. Of course, you're having an effect on the cells immediately around there, but you're mainly stimulating the white matter. So in Helen's case, she was always stimulating the white matter that was just adjacent to areas 25 and 32. So it would have been the uncinate fasciculus. And now she's particularly focused on the area that brings uncinate fasciculus and the corpus callosum, the ventral part of the corpus callosum together. So yeah, it's mainly the white matter. The cingulotomies, no, they're much more caudal and dorsal. So she's in the ventral part of the subgenual, and the cingulotomies are really dorsal anterior cingulate and more caudal. And those are getting the cingulate cortex and the cingulum bundle. They don't go quite to the midline, which is one of the things that we're interested in seeing what is spared, because they can't go to the midline for safety reasons. So you're always going to be sparing some of those fibers. And one of the things we've been quite interested in is understanding how well and which ones are spared. Right, so that's exactly what would be, of course, important to probe in a rodent or, a marmoset model. I think it depends on what comes into a given area. So the place that I had shown that parietal cortex comes in, and remember there's, transmitter systems there that are, modulating the behavior all the time. So that's an area that gets input. That particular one would get input from VLPFC and OFC. And OFC is important. So we were thinking about it in terms of being able to direct your attention, because you get this parietal cortical input and dorsolateral prefrontal cortex, to some very salient important stimuli in which you have a particular goal. So that would involve the orbital and dorsolateral and parietal cortex. And so one of the areas that we'd be interested in, for example, is in addicted individuals. We know that as people become more addicted, and in animals, the activity moves pretty much from the ventral parts of the striatum to the more dorsal parts of the striatum. So one question one could ask then is this area, along with its dopamine input, et cetera, particularly important in sort of stamping in those habits that are very hard to break? Just an example of how one might think about the different combinations of inputs that might come into different parts of the striatum. Yeah, so that's a really interesting question. I don't know, but what I do know is that the cortical thalamic cortical connection does have that attribute. So we also looked at that some time ago. So the cortex projects to the thalamus in a wider area than thalamus projects to the cortex. And the thalamocortical projection is much more precise. So there is this shifting that does occur. And it actually, we were surprised to find, but it actually occurs in the same direction. We were thinking maybe it would occur in the reverse direction, but it doesn't. What we're doing now is looking at those downstream projections from striatum to pallidum, and then pallidum actually to the STN, because we want to ask a similar question, which is how is information to the striatum transferred then to the pallidum? And that projection is very topographic. So once you have this organization of convergence and potential integration in the striatum, that whatever processing that's doing comes down to the pallidum, and there's something else going down there, but that is very topographic. You don't have the integration so much from those pathways. Thank you.